Last December, a tumultuous commotion took the country by storm regarding the smuggling of illegal meat cartel. A warehouse in Senai, Johor was being raided by Malaysian Quarantine and Inspection Service (MAQIS). The warehouse was believed to be the cartel of illegal meat smuggling and distributed to the market nationwide. This cartel printed fake Jakim Halal logo and stickers after repacking the meats imported from China, Ukraine, Brazil, and Argentina. According to the reporter, the New Straits Times newspaper, the information obtained after conducting thorough research was the meats were suspected of having dubious quality since it was mixed with low-quality meats besides horse and kangaroo meat were being included. This issue also tarnished the good name and reputation of some Malaysian authorities such as Department of Islamic Development Malaysia. The objective of this video is to unfold the analysis method which can be applied in this food controversy issue. The mixing of horse and kangaroo meat issue was being raised the controversy of illegal meat cartel, and this issue was being the talk of the town since horse meat was not allowed to enter Malaysian market. Ministry of Domestic Trade and Consumer Affairs (KPD and HEP) certified that no horse, kangaroo, or pork meat was found following the seizure in the warehouse. However, most consumers were still hesitant to consume the meat due to the impact of fake halal logo issue that was being practiced in that warehouse for 40 years. A method that can separate DNA fragment or other micromolecules such as RNA and protein based on their size and charge is called gel electrophoresis. These separation techniques are based on the movement of charged molecule when exposed to an electrical field, and this movement occurs in a gel medium that generally used was agarose that obtained from the seaweed.
So what is gel electrophoresis? It is a technique used to separate charged molecules on the basis of size and charge. Nucleic acid can be separated as whole chromosomes or as fragments. The nucleic acids are loaded into a slot into one end of a gel matrix, and electric current is applied, and negatively charged molecules are put toward the opposite end of the gel, which is the end with the positive electrode. So based on the size of the molecules, it will travel through the gel at different speed, which allow them to separate each other. It also can be based on their DNA chart if there is any difference in charge among them. In case of DNA, all molecules have the same charge to mass ratio, which they are negatively charged. The separation also depends on the compactness of the team, where the more compact the DNA, the faster it will bite right through the gel, where the smaller molecules move through the pores in the gel faster than the larger molecules. So this difference in the rate of migration separates the fragments on the basis of the size. The nucleic acids in a gel matrix are invisible until they are stained with a compound that allows them to be seen, such as dye. Distinct fragments of nucleic acid will appear as bands at specific distances from top of the gel, which is the negative electrode end, that are based on the size. A mixture of many fragments, varying sizes appear as a long smear, whereas uncut genomic DNA is usually too large to run through the gel and forms a single large band at the top of the gel.
The function of gel electrophoresis in solving or identifying the presence of horse and kangaroo meat's DNA can be strengthened by the case study that I read, which is detection of pork DNA in processed meat products collected from self-proclaimed halal Muslim street food stalls in Hat Yai, Thailand. 36 samples of various processed meat products were randomly collected from 7 Muslim street food stalls. Next, the DNA of the samples were extracted. Then DNA products were amplified by PCR and analyzed by agarose gel electrophoresis. This showed that gel electrophoresis is an important step for validation, as the results of a PCR reaction are usually visualized using gel electrophoresis. It is a technique in which fragments of DNA are pulled through a gel matrix by an electric current and separates DNA fragments according to size. A standard or DNA ladder is typically included so that the size of the fragments in the PCR sample can be dated. The results showed that 3 out of 36 samples were positively identified to contain porcine DNA, and it can be concluded that the DNA method conveniently provides reliable results for routine food analysis of halal requirement, as this study highlights the importance of halal integrity between producers, suppliers, and street food business owners to provide halal food products, particularly to Muslim consumers.
Therefore, as in the case of this illegal import meat cartel, the smuggling of meat was due to the manufacturer who did not obtain JAKIM approval since they failed to comply the halal certificate standard, and they took shortcut to obtain profit by successfully breaking through the government fortifications. Our objective is to give exposure towards this instrument, as we would like to urge meat consumers, especially small business owners and restaurants that use meat in their dish, to be more cautious of the meats used. We would like to suggest them to send the meats being used in their food periodically to Department of Chemistry Malaysia to assure the meats had no mixture of other meats. Besides also ensuring the safety of the meat, and they can exhibit the result in their premise to assure their customers. The Department of Chemistry Malaysia consists of nationwide network of laboratories that provide analysis, investigation, and scientific consultation to government agencies. In addition, business owners also can send their samples of meat to private laboratories recognized by the Ministry of Health Malaysia for food analysis service. The list can be found on the official website of the Food Safety and Quality Divisions of the Ministry of Health Malaysia. That's all from us. We hope that our video will help consumers to be more aware in their food selection, especially regarding halal status.